right, hi everybody. We're going to be going over some Spectronaut files today. This is how we usually distribute data using the software we use for DIA. So I thought I'd give you guys a little tip on opening your data, looking at it. I haven't looked at this data in a while, so we'll be doing it together. So we'll see how we do. All of these files, you can download them or not, it doesn't matter. But all of them could be found on this website here, bioshare.bioinformatics.ucdavis.edu/bioshare/view/Spectronaut. And feel free to pause the video at any time. I go kind of fast, but since it's a video, you guys can pause it and knock yourself out and rewind it and do all sorts of things. So if you want to download this data, great. If not, just follow along. We'll be looking at this file right at the bottom, the one 3.5 gigabytes that says 50 nanograms DIA QC runs DIA search. So this is just some 50 nanogram K562 files we ran as a QC. We usually run between 50 and 200 nanograms. And I believe this file has some different gradient lengths in it. So please feel free to download it or not. But when you do download it, you will see, let's see where I already downloaded it already, and where is it? Let's see, I think I put it under Spectronaut. And so you can, once you download the viewer, so you will need a Spectronaut viewer. So unfortunately, you just can't download it from their website, but you can, if we've given you data in Spectronaut, email me, or I usually email you or someone in the core and give you just instructions on how to download the viewer. If you don't have data from me, I think you can ask them. I don't know, they're very nice. You can ask for a viewer to open this if you want. So I think we're going to open this one. Actually, I think we're going to open this one, and I will put this on the BioShare. So actually look for this one, which is 4.8 gigabytes. No. Let's do that later. Let's do this one. So the one we originally planned, the 50 nanogram QC, this is the one we fixed some of the data. So we'll just double click on this. Once you download it, you'll see Spectronaut open. It usually takes a couple minutes to open the data, and let's see. Okay, so it's starting to load. These are all the mass spectrometry files that are over in the upper left. I'll kind of give you an overview of the software as it happens. You can see it takes a couple minutes to open the file. Sometimes it could take as long as 10 minutes, maybe not 10, maybe like 5. But if you have like 100 files in here, maybe it can take I don't know, like 10 minutes or so. Also, they do not have a viewer for Linux or a Mac, so if you have one of those, you're kind of out of luck. I believe you can probably get it by a computer. I know it's kind of unfortunate. Or like something in the cloud might work. I probably wouldn't recommend it, but if you want to look at your Spectronaut data, you're going to have to use a Windows box as far as I know. Okay, so as long as when this finishes loading, you'll see this finished loading the file. Okay, so let's go over what some of these views are. You'll kind of see these panes. There's analysis, post-analysis, report, database, and settings. We're just going to be going over analysis and post-analysis. It kind of defaults to this tree view. So I'm using the viewer just like you will. So I'm not even using the full software, although it does almost everything the full software could do. You could see the different files that are loaded. There's currently 1, 2, 3, 4, 5, 6, 7, 8 mass spectrometry LCMS files loaded. They're red because they are missing their raw files. Usually this does not make any difference, but there's some visualizations where it's still needed. I still haven't quite figured out what those are. If you want to turn them not red, you could download all of these here, or I believe we've already converted these to Spectronaut HTRMS file, and we have the converter to do this, so don't worry about it. But you could also load the raw data or the converted files. Okay, So most of the time you don't need the raw data, but there are a couple visualizations where you'll need it. So let's start out really quickly at the view it defaults with. It defaults using on the upper left panel the IRT calibration and the lower panel the analysis summary. You could change these by doing the drop-down box. We'll go through some of these today. So I can explain IRT really quick. I probably should, since it's like the first video to pop up. I mean, the first visualization to pop up. So what happens is, and I will explain kind of what DIA data is, and there's some videos before this we made last year that kind of explain it. But all of these are the peptides that we're identifying in the run. It converts the retention time. So remember, we're doing LCMS, so that's peptides are eluting on an LC column over time. So instead of a physical time, which would be here on the left-hand corner, the y-axis, which would be retention time in minutes. You could see this run is only 30 minutes long. It can convert the peptides into what's called IRT space. So you can think of this as like goal posting. So like it uses a deep learning algorithm that's much better than the algorithms that people used to use like 5 or 6 years ago to take the sequence of peptides and kind of predict what the retention time is going to be. But because they don't know what our gradient length is, it has to convert it into an IRT space. So basically what this does is it takes the peptides that are known to be most hydrophobic, that is eluting most late in the run, so eluting around 25 minutes of the 30-minute total time, and gives it a number. So like really hydrophobic peptides are going to be like 160. It looks like it's around 160. And that goes all the way down to really hydrophilic peptides, which are given a number. I think it looks like it goes down to negative 100, negative 60. So it's kind of like goal posting it. So it takes all the real hydrophobic ones that are predicted to be hydrophobic by the amino acid sequence and gives it the top the top numbers of 150. And then it gives the least hydrophobic ones a negative number, and then does a regression, a linear fit of your peptides as they go through time from most hydrophobic to least hydrophobic. And then so what this is a graph of is the prediction of the IRT peptides related to the actual retention time. So this is how the predicted retention times match the physical ones. And so what this allows you to do is, for example, we have different runs with different gradient lengths here. This is very unusual. So like when we run your runs, we have the same gradient length for every single run. But in this case, we have different ones. We have some at 25 minutes, 45 minutes, 60, and 90. So we're kind of measuring, trying to find the sweet spot between number of IDs and number of trying to do your runs as fast as possible, right? Because you pay per time. So the faster we can run the runs, the less it'll cost you. So like, for example, this 25-minute run is going to cost you half as much as the 60-minute. So if we get most of our data in the 25-minute runs, you have to pay half as much. So that's good. Yay. So this is kind of what we're looking at here. So basically just a prediction of how the hydrophobic peptides match with reality. And it looks really good. And you can see these curves aren't exactly linear, which we see all the time. So it's not strictly a linear regression. It's some other sort of regression. Okay, at the bottom we have the analysis summary. So right here is the number of precursors identified. And if you go over these, it'll give you like a tooltip of kind of what they mean. You could also go under About and download the user manual. But a lot of this stuff is more explained in the tooltips in the manual, although the manual is really good for a manual. So anyway, what these are telling you is there's 3 numbers here for precursors. So we should step back and figure out what a precursor is. So peptides can exist as multiple precursors. That's why there's more precursors than peptides, for example. In this run, there's 50,000 precursors and 46,000 peptides identified. That's really good for 30 minutes for 50 nanograms. So that's pretty awesome. So anyway, this is the numbers that aren't in parentheses are the numbers for this experiment, and the numbers in parentheses are the total, I believe, for the whole experiment. Okay, so 51,000 precursors. You can kind of ignore modified peptides for this. Peptides, 46,000. Protein groups, 5,000. So we identified 5,000 protein groups for this one run in 25 minutes for 50 nanograms. That's pretty awesome. This is the average peptides per protein group. So the average number of peptides identified per protein group. So what is a protein group? So remember, we're looking at peptides. So a lot of those peptides can be in multiple proteins. And sometimes when we only have 2 or 3 peptides, all of those peptides can be in multiple proteins. Then we say we only identified a protein group instead of an individual protein. This could be important because if you're looking for an individual protein, and there's a highly homologous class of proteins, you may not know exactly what protein we've identified and quantified here. So keep that in mind. So average number of peptides per protein group, precursors. I usually ignore precursors. Average miscleavages. So this is the how well our enzyme did. This is 20%, and our enzyme did not do very good. But this is a standard we bought from Promega, so you could blame Promega. So the number of miscleavages, 14%. So yeah, around 14%. Okay. What are some of these are really geeky, nerdy kind of things here you can ignore. These you can all safely ignore. Let's see, LC capacity, you can ignore this. Cycle time is important. So we'll go over cycle time, and maybe this video, I may cut this video a little short and do this in sections, but cycle time is important. The lower the cycle time, the better. Let's see, what else? Tells you this is a TOF, so this was analyzed on our TIMS TOF. Kind of other geeky things here. Tells you the date, the vendor, the instrument. Actually has a serial number, so you can go back and see what the serial number is. Okay, and as you move, you can see this changing, right? So like, look at peptides. So in peptides, in this file, we identified 46,000. In this one, 50,000. So this one did a much better. And you can see raw files are missing. Some plots may not function properly. If this happens, you click this run and go map raw files. And then you download those files from the FTP site. We go to the data. I downloaded them on data. Let's see, where's Spectronaut, I believe. Spectronaut, where is it? There we go. And here's all the files here. You just hit OK, and it should map them. Yes, and so they turn from red to X's to green. So very simple. Okay, and you get OK, boom. All right, so now they're all not red anymore. Yay! So if we don't give you, so sometimes we don't give the raw data files to people, and we don't do that on purpose. It's just because they're enormous, and it takes a lot of time to download and to upload, and it takes a lot of time for you to download. And for TIMS TOFs, they're kind of anyway, it's a pain. But we'd be glad to give them to you. They're yours. You paid for them. If you need them, let us know. But for the most part, you don't. But I'm always happy to give them to people. I mean, I really think you should have them because there's other programs you can use them for. Just give yourself a couple days to download them. If you're under a slow internet connection, I believe this is about 40 gigabytes for 1, 2, 3, 4, 5, 6, 7, 8 files or so. Okay. Let's look at some other graphs. So one of the reasons we really like Spectronaut is because the visualization is really awesome. All of these you can kind of ignore. They're like you don't have to ignore them. They're fun to play with. For example, you could put XIC extraction width. What does that mean? Well, a lot of these are kind of hard to explain. But an XIC is basically the region of the chromatogram it's extracting data from. And so you can see based on the retention time prediction, basically this is the width around the retention time that it's looking for this signal. And you could see how that changes over the gradient depending on how the retention time is accurately mapped. So it looks like you're doing about Oh, I don't know. That's time in minutes, so a minute or two. So that's pretty good. So the tighter your predicted retention time, the more accurate your extraction width is going to be. So that's kind of all you're looking at here. And you can see that changes across. This is a different gradient, so I don't know why that looks like that. Sometimes I think you have to scale the default. There you go. That looks better. So when we change gradient lengths, you could kind of see. So we've moved from like a 25 to 45 minute gradient. You could kind of see how their prediction kind of matches pretty good here, but the curve looks a little different. Linear retention time prediction, high accuracy retention time prediction. Why wouldn't you want to use high accuracy? I don't know. It seems like you should always use high accuracy. Anyway, that kind of, it's interesting to look at. What I like is if there's timsTOF data, well, even if you do Orbitrap data, the base peak chromatogram is good to look at. I believe you do need the raw data for this. That's probably one of those things you need the raw data for, because it says loading data from raw files. So it's looking for those raw files we gave it. It can take a while. So let's leave that and go to ion mobility overview. Oh, that's pretty. I like this. So the timsTOF is kind of an dual ion mobility, trapped ion mobility mass spectrometer. So there's actually 4 dimensions of data. So Spectronaut uses both the ion mobility, which is basically the shape. So think of peptides going through a wind tunnel, right? And based on how their shape is, they will go through this tunnel at like different speeds, more or less. It's kind of different when it's trapped ion mobility, but it has to do with how the peptide is kind of shaped. So even if the peptide has the same mass, but like a different positional isomer, like let's say there's a serine or threonine or 2 serines next to each other that are phosphorylated. If it's on one serine or the one adjacent to it, it may have a different retention time. Not a different retention time. Well, they might have a different retention time, but they should have a different ion mobility. So you can use both the retention time and the ion mobility to kind of determine if the positional isomers are correct. So I'll show you some phosphorylation data on that later. Spectronaut is awesome for phosphorylation and ubiquitination and several other modifications. So what are we looking at here? So this is 4-dimensional data. On the x-axis, we got the mass over charge, right? We all know what mass over charge is. On the y-axis, we got ion mobility, and now we're like searching them. We're looking at the data like from the top. So the heat map is kind of the intensity of the signals. And these red boxes are the ion mobility windows. So these are kind of the, not the ion mobility, the data independent acquisition windows. So go look at our data independent acquisition talk, which I really have to redo. But this will kind of tell you what the DIA windows are. And you could see there's dots outside the windows. So if anything's outside the windows, we won't see it. So this is not normally important for these type of experiments because they're bottom-up digested experiments. But if you're looking for cross-linking or you're looking for something big, you may want to double check because anything outside of these windows, like these right here, we will not see. So why do we have 2 clouds here? It turns out these are the +1s and these are the multiply charged peptides. So we don't normally care about +1 peptides because they don't fragment well. And usually for bottom-up proteomics, we only concentrate on the multiply charged peptides. But you can see there's like kind of some dots here, and these will not be retained in the analysis. So it's kind of good to check where all of your data is coming out. And in this case, it looks like we're sampling most of it. So usually that's not that important. You can look at the mass calibration, ion mobility calibration. You kind of see, how the data is calibrated. It's kind of geeky things you can look at if you like to look at mass spectrometry data. MS1, MS2, tick chromatogram, ion mobility extraction width. So this is on the ion mobility extraction. So this is what the error rate on or at least the error, not the error, but where it looks at, where most of the data is between the ion mobility. So what can we do here? Some of the things we like to do to get you started. You can play around with this geeky stuff, which I really like to do, and you totally should if you want to. You could also detach these windows. Like, for example, you can detach the ion mobility extraction width, and it will actually show up as another window. So you can have all sorts of windows if you have multiple monitors or whatever. It makes it really nice. Okay, so for us biologists, probably the thing you want to look at most is the grid view. So this will tell you there's some changes I like to make to this. So please pause and rewind. You rewind a video? I don't know, not really. So what I like to do is I like to add a couple columns for this. So this is all the proteins you've identified, all 5,000 or whatever they are. Let's see. The completeness, so this is how many times this protein was found in all the runs. The coefficient of variation, so this is a measurement of how accurately we can measure this protein across runs. If the protein's changing, expect the CV to be higher. And these are the different runs we're looking at here. So we have 2 at 25 minutes, 2 at 45, 2 at 60, and 2 at 90. I actually have a video on this on the Spectronaut website. I think it's on YouTube if you want to see it. I kind of went through this data pretty quickly. But one of the things I like to do is you right click, I believe here, there's lots of, so in mass spec, like, I don't know if this is the same more software, but in terms of mass spectrometry software, there's always hidden menus. So always try to right click anywhere you can, and you'll almost always find hidden menus. Okay, so we're going to right click, we're going to say column chooser, and we're going to choose some other columns that are important to us biologists. And why they don't default to this, I have no idea. But I would default to this. Okay, so we're going to put number of peptides, which I really like. And we're going to look at protein description. So this will tell us a little bit more about what these proteins are. Okay, awesome. And if we do differential expression, there's like a candidate here, and that means this protein was found to be differentially expressed across the runs. So why are they differentially expressed across the runs if they're the exact same sample? It's because we have different gradient lengths. And it's actually really interesting, this data, so we can go over that later. Here's the coverage. So here's the coverage, what peptides you found. So basically, you've seen this before. This is like the N-terminus of the peptide. This is the C-terminus. We digest it all with trypsin. And these are the peptides we find. Okay. So what I like to do is sort by the number of peptides. So generally, the more abundant a protein is, the more peptides you're going to find. It's not exactly the case most of the time, but for this purposes, it's a good measure. So we can sort by number of peptides now. So these are the most abundant proteins we're finding in these K562 standards we use all the time. So this one, all the ones you expect, plectin. I don't know what this one is. What is this one? I don't think I've seen this one before. That's interesting. Probably someone seeing this video are like, oh yeah, I know exactly what that is. But let's look at it for a second. Okay, Medicin. You can look at the accession number, look it up in Uniprot if you want. Okay, so we've identified 150 peptides. Pretty awesome. Completeness is 100%, of course, because it's very abundant. You would expect to see it on all your replicate runs. CV is pretty low. So usually the CV with cell culture, we expect, around 15 to 20%, maybe a little bit less than 20%. These are standards, so the CVs are going to be low, but the CV will vary by protein. Okay. So we have little bars that are green. That means we have identified this protein, this peptide in this protein. Bars that are yellow, this means we maybe identified it. We're not too sure. It's basically a Q value over 5%. This is a Q value under 1%. So we kind of filter at stuff under 1%. And what this means, I'll show you later. We could go over this for days, which we probably will. Anyway, just for now, let's think of the green as really, really good evidence. The yellow is maybe iffy, and the red's like, no, this isn't good, but we're showing you it anyway because it may still exist, but it just might be crappy. There might be not enough data to look at it. Okay, So what we can do on the bottom, we have profile. Thankfully, we can change this now, so it defaults to log 2. So remember, this is, so if it's a log change, log 2 change, it may look the same when there's actually like major differences, right? So I like to view this. So remember, right click. The secret is always right click on stuff. You will find secret things. Unclick, use the log 2 scale, and then you could kind of see the difference more generally. So you could see this protein, the software think is more highly expressed in the 25 minutes. This is interesting. One of these 45-minute runs didn't work very well. And then you could see that seems to be less expressed in the longer runs. Why is that? Well, that's interesting. It's probably because the peaks are higher. I don't know exactly. That's my theory anyway, but they should be the same. But remember, with your data, we use the same gradient throughout. So with ours, with this experiment, we're doing different gradients, which we would never do with your data. So, but it's kind of an interesting analytical exercise to try to guess why this protein seems to be differentially expressed with more expressed with a shorter gradient than with a longer one. If you guys have any thoughts on this, I have some theories. Just let me know. So we'll go through what transitions look like, but if you want to click on these numbers, you could kind of look at the data. And this is kind of what DIA data will look like for this protein, for example, in this run. So you can kind of see this one. And as you go through runs, you can see some that don't look very good. You could see this one, this 45-minute run has lots of reds. This run did not work very well. It might have been like an autosampler issue or something. This one looked a lot better, and I'll show you what good data should look like. Some of the data might look like this. That might be because we have imputation turned on, and I will show you how to turn that off and what that means. In one of these videos, there'll probably be like 4 of these videos. Okay, so basically this is to get you started. This will show you the number of the proteins, how they're distributed across your different runs, whether they're differentially expressed, and for what proteins. So kind of click on the name to get the bar, click on the numbers to look at the DIA data. If you see a lot of stuff that looks like background, that's probably because we have imputation on, and we're going to show you how to turn that off. And I will show you what really good data looks like and what kind of crappy data looks like. Okay, so we're going to go back to the tree view. I think that's going to get you started for now. But let's look through, let's give you one. I'm trying to decide how we're going to organize this because I'm just kind of doing it on the fly. So let's go over, before we look at the post-analysis, let's continue on the analysis. So if we right-click and look at one of our hidden menus again, we'll see add runs. You can add runs. You can if you're a viewer, but you can if you have the real software. You can add a run. So, but you guys can remove runs. You could order runs by acquisition date, which is good. So you can kind of see if, like, we did something stupid, like we ran all the controls before the experimentals, which we don't do. But if someone did, you can sort by acquisition date. Like you can see if we sort by acquisition date, you can see I've randomized these. So I do 25, 45, 60, 90, 20. I didn't actually randomize them, so it looks like I did 25, 45, 60, 90, 25, 45, 60, 90. And one of the things we do on the timsTOF, well, it'll assign in a number. So you can go by this number, and that'll also tell you the order. So 7, 13, 17, 19, 21, 23, 27, 29, 731. Orbitrap data doesn't do this, unfortunately. That's pretty cool. I actually forgot I ran them like this, or we ran them like this. I think Gabriella ran it. She did a great job. You can also order runs by original order, which is 25, 45, 60, 90. Order runs by condition replicate, analysis date, group by. We'll go through group by later. Okay, so we've ordered by condition 25, 45, 60, 90. If you click on this little arrow, it defaults to the windows. So these are these DIA windows that we saw on the ion mobility overview, which you can see right here again. You can see these windows, and that's what these windows are. So you got like 457 to 482, that would be like this one maybe, and like 482 to 507, that would be this one and that one. And this is useful for a mass spectrometrist. It's not so useful for a biologist. So we can change the grouping. But for us, we could keep it like this for now. And you can see there's different levels of like trees here. So we click on this one, and this will tell us the peptides we've identified in this precursor window. And so if you hover over the peptide, it'll tell you a little bit about it, what database it is, the protein name, peptide, gene, all sorts of things. If you click on this again, it'll tell you the mass over charge of the precursor. You can see this makes sense. So it's 458. It goes in, it fits between 457 and 482, which it should. Click on it again, and it tells you what the fragmentation products we found. So let's just click on this and see what it looks like. Hopefully it'll look good. Maybe, yes, it does. Okay, it does look good. So one of the first things you'll notice is there's not enough points across the peak. You kind of see this as a triangle. This was our 25-minute run before we fixed it. And so we've actually decreased the cycle time in half. This is why cycle time is important. So especially with this LC setup, we have very narrow peaks, maybe like 3 seconds, 3 to 6 seconds. And so the instrument only has so much time to assay it. And so you want to have at least 6, maybe 11 points across the peak. You can see we only have like 3 here, so it kind of looks like a triangle. So the quantitative accuracy of this is not going to be very good. So when you look at DIA data, kind of look at how your peaks look. If they look like a triangle, well, your quantitative accuracy is not going to be very good. It's going to be okay. It's not great. Okay. So what we're looking at here, the upper panel is actually the MS2, which is the MSMS fragmentation products from the precursor, which is at the bottom. And the MSMS is usually more important, and that's why we put it on top, or why it defaults on the top. In the lower panel is the MS1. Okay, so go back and look at my DIA video to see what this actually means. But essentially, if you look here, these are the masses. This is the M+1 monoisotopic. So actually the isotopes. So monoisotopic mass, the mass with one isotope, 2 isotopes, 3 isotopes. And you can see there's kind of a pattern. Here's the predicted retention time. That's here. This is the actual retention time. So you can see we have very good prediction between the very good alignment between the predicted retention time and the real retention time. So this is the intact peptide. These are the masses of the fragments, the Y ions or B ions. And so you guys go back and look at the, if you're kind of fuzzy on what Y ions or B ions are, go back to one of my original videos, and hopefully I explained it okay. If not, I hope to explain it a little better. And you can see these kind of line up. So this is how DIA works, especially without a library. You can kind of see how we're kind of identifying this as being real, because these fragment ion products align with the precursor product. And this is one of the tests Spectronaut uses to see if it's identified correctly. So alignment with retention time, ratio, spectral angle. I'll explain what that is in a minute. Whether they co-elutant time. This is all how we identify DIA data. It's actually quite remarkable. So let's add a new window. And so what we're going to do is we're going to look at, let's see, data point match. I like this one. This is super cool. So let's look at all of these aligned together. So here is your intensity of your precursor. You can see this is 458, whatever I remember, 458.29. This is what the precursors look like, the M+0, M+1, M+2. These are these isotopes I talked about in a different video. They should have a pattern that is predictable, and you could see kind of how those match in a different plot, which I will show you. Here is the MS-MS spectra. So you can see DIA spectra are very, very complex. So remember, MS-MS spectra from DIA contain hundreds, maybe, of peptides. Yeah, probably hundreds. So it kind of pulled these out of the spectra, right? Because these are the ones it's predicting, the deep learning prediction should have from this peptide. It finds them in the spectra. And Spectronaut says, hey, do these colute? They do colute, which is upper panel. They all colute at the same time. That's this Y6543. You can see that here. Y6, Y5, Y4, Y3. They all colute at the same time, right? And here's the MS, the M0, M1, M2. You can see those also elute at the same time, and they elute at the same time with each other, the MS to MS-MS. This is how DIA works. It's awesome. Okay, we can also look at, let's see, what's the MS1 ion mobilogram? So this is the predicted ion mobility, that kind of fourth dimensional data. You can see it matches. So this is the prediction. This is the real ion mobility. So it predicted it to be around 0.87. It's actually about 0.90. Is that good? I don't know. I think it's good. Here's the MS-MS, how they elute. And remember, if these fragments all come from the same precursor, they should all have the same ion mobility. Let's see, what else we can look at. We can check the MS2 intensity correlation. And so what that is, remember these Y ions we predicted, like Y3, 4, 5, 5 minus water, 5, and 6. This is what we see in our mass spectrometer on the top. This is what our deep learning predictions say we should have. And the intensity relative to each other is important because the deep learning prediction can predict that too. So this is new within the last, really practically new within the last 5 years. So the difference between these angles between 5 and 6, people call this a spectral angle. So we know the deep learning prediction, for example, if you look at Y5 and Y6 fragments, should have an angle associated with their apexes, I assume. And that angle should match what we see in our mass spectrometer. So for example, we're predicting Y6 is less intense than Y5. Let's see, the predicted's on the bottom. If you take their apex, there should be an angle associated with that. I don't actually know where they start the measuring the angle. Maybe this is like 10 degrees, I don't know. But it should be the same. And it is. So that's one of the ways we actually identify stuff from really, really complex spectrum. Okay. So MS2 intensity correlation, MS2 isotope intensity correlation. Okay, that's not very as useful. What else? You can actually, we'll look at stuff across runs later, but right now let's just look at this. Okay, so these all correlate together. This is a good ID. You could tell by the Q value, which is low. We've seen some like minus 30, but minus 5 is a good Q value. And you could just look at the data and it looks great. Okay. You can also, there's all sorts of right-click craziness you can do. You could change this to use IRT scale. I don't know why you'd want to, but you could. Let's see, use normalized intensities. Usually we normalize things as a talk for another day. You could use the full range XIC to kind of, this is funny, like when you do this, you can kind of see it's like, whoa, look at how noisy that is. And when you look at that, you're like, whoa, this is super noisy. But look at how beautiful it is. It's just beautiful. And what happened to our other window? It's like this. Oh, we have it both. Let's see. Yeah. Okay. So that looks good. You can also, there's usually interference, remove interference, but I don't see that here. So we can look at peptides grouped per protein. You can see, let's see, grouped for precursor range. So here's another one, and we can set this as run to XIC, reg1 XIC. You can see this is a little lower in intensity. However, the Q value is very good, so there has some things too. And you can see it's kind of not as many points across the peak as I would like. So this is why we stopped using this method. Let's see. Okay, so you can look at other precursor ranges like this one. Ooh, that looks pretty. You could see the retention time. It looks a little bit far off, but see we're still zoomed in. So like it's predicting it to be 10 minutes. And because this is an analytical software, it's 100th of a second. So 10.2 versus 10.4. So that's not bad. Okay. So we can group this by precursor range. If you're an analytical chemist or a mass spectrometrist, this is cool. You're a biologist, not so useful. Unless you're looking for, oh, that's cool. You can look at all of them here. Oh, that's neat. And you can see some of them don't look some of them look good. These don't. That might be imputed. I'll explain that in a different video. We're kind of running out of time. But what I want to show you today is right click, say, we want to right click somewhere down here. Group by, we could do protein ID. I like that one. That one's kind of cool. So now when you open this, they'll be grouped by protein. So if you know your favorite protein, like this one, whatever this one is, you can click on it. It'll have your coverage at the bottom and your data across all the peptides here. Kind of cool. Let's see. So we can look at this one. Yeah, you can see. It doesn't look very good. The data looks good. It's definitely identified, but you can see we don't have enough points across the peak. What's cool is this let's see if it does its magic. MS2 alignment across runs, cross-run RT accuracy. All right, that's cool. You can see there's something wrong with this 45 minutes. Yeah, this is just amazing. So why this is amazing is that what we're looking at here, and this is one of my favorite plots, is how this is sampled across different runs. So we have, how many runs do we have again? We have 2 at 25, 2 at 45, 2 at 60, and 2 at 90. So because of the IRT space and the ability to warp peptides across different gradients, it knows exactly. So it'll elute at a 25-minute gradient at a certain time. That should be a lot different if you have a 45-minute, 60, or 90, right? Because it may elute at 5 minutes here. If you have a 45-minute run, it may elute at, 15 minutes. At a 60-minute run, it should elute at maybe 20, 50, I don't know. And then 90-minute run, maybe it elutes at 75, I don't know. So, but because it's all translated into IRT space and mapped together, you can map them across different gradients. This is actually really cool. I don't know why you'd want to run a sample at different gradients, but you can actually see it, make sense out of it, and you can see there's something wrong with this run. But it's just amazing. Like, it is just absolutely amazing how it knows where this peptide is across different gradients. It's very, very super cool. And you can look at this XIC graph, which tells you kind of the same thing. So this is the signal in all the different runs. So like 25 minutes, 45 minutes, 60 and 90. And these are the area under the peak for, you can see the 25 minutes, 45, 60, and 90. And you can see how this looks. See how this looks. Let's look at this one. Let's see if we can click on that. Oh, does that work? Okay, so look at the points across the peak here. So you can see it kind of looks like a triangle. So we're not getting a lot of points across the peak from this peptide. You go all the way over to 90 and you expect more. Yeah, and you can see it looks more like a curve. It doesn't look great, but it certainly doesn't look like a triangle. So the longer the gradients, the more points we get across the peak. We don't necessarily identify more peptides, although we'll check that out later. The data is very fascinating. But remember, this 60-minute run is going to cost you twice as much as the 25-minute run because we charge by time. So you get more quantitative accuracy the longer your gradient, but you're going to pay for it. Unfortunately, because these machines are like super expensive. So like I just paid the service contract for this, and so this is why we charge per time. But super cool what we can do. Oh, and one thing I should show you is that this is actually really cool. It may be explained somewhere in the manual, may not, but you can see if we zoom in on this, how some are blue and some are transitions are gray. The blue means we're using them. The gray means I think they're interfered. So because these signals are so complex, sometimes you get transitions that aren't very diagnostic for your peak. So Spectronaut will figure that out and remove them for you. I believe there is a way, if you want to use them, you could turn it back on or turn them off. I think there is a use for quantitation. So for example, if we wanted to use this one, we could turn it back on to say use for quantitation. So Spectronaut thinks we don't need it, so I tend to believe Spectronaut. But if you wanted to, you can say, hey, turn this back on, and then it will turn it on. And what can turn on this one? Boom, turn that on. We can turn that on. So we can use more of them if you want. It may make the data better. It may make it worse. We can check by looking at the cross runs XIC graph. Did that make it better? Did that make it worse? I don't know. It looks better to me, but I don't know. We'll see. So anyway, that's it for now. So remember to group by peptide signal, add your protein description, number of peptides. Remember to change this from log scale to not use log scale. I don't like log scale for this chart, but you may. Some people really like to look at log scale data. So if you like log scale data, knock yourself out. I don't. Ooh, look, there's a shooting protein. Shooting 1. Awesome. Look at that. So here's all the peptides we identified from shooting 1. You can kind of see those. Some look good, some don't. And I'll go into why you get data like this and how it might be imputed and how to tell and how to change your settings in another video. But we've been doing this for how long now? I don't even know. 47 minutes, I think. Wow. All right. So we're going to stop here. And then the next video, we're going to look at the post-analysis, maybe how to change. Oh, we're going to go over changing your settings with another secret menu. Go over the post-analysis, kind of get you started. I really encourage people to just look at their data and spend like hours and hours and hours of time looking at it, because it's really fun. And actually you can find stuff like in your data that like we don't see, just because you have more time to look at it. Okay, isn't it amazing how it picks this out of the grass? It's just amazing. So like we can go back and look at the peptide data match, and I bet it's going to be in the grass. We'll see. Almost certainly will. Oh yeah, look at that. That's just awesome. So like, let's see. Yeah, look at that. So 1, 2, 3, 4, 5, 6 transitions. We use 3 out of the 6. You can actually zoom in here and see. Oh, God, look at that. I mean, it picked this out of the grass. And you're like, how can this possibly be real? Oh, we must have something clicked here. Extended library fragments. Let's see. Yeah, there we go. So here's the stuff it picked out of the grass. Isn't that, it's just absolutely amazing that I can pick this out of this complex data. And like, how could that possibly be real? Like, but a lot of times I believe it. Most of the time I do. Because you can see how these fragments that it picked out of the grass align in time. It matches with the MS. This is noisy, but you can see the MS signal clearly. And then you can pick out the MS-MS fragmentation. And what Spectronaut does, which is really nice, is it uses both the MS and the MS-MS for fragmentation. So you might not want, for quantitation, so you may not want to use this, but it kind of scales them. And you can view this across runs, right? So you can go to the cross runs. I'm just screwing around now at the end of the video. XIC graph. There you go. Beautiful. This is a real signal. So this is this peptide across multiple runs. Looks totally real. And you can see how the dotted ones are the interfering ones. You can see how these don't exactly line up. That's why they're probably interfering. So like this blue one, for example, seems like it lines up here, but on this one it doesn't. So I think it actually looks at interference across all your runs and not just your individual runs. So turning on or off interfering peptides may be a good thing for one sample, but might not be good for quantifying it across multiple samples. That's cool. Really cool. DIA is awesome. Okay, so I'm going to sign off for now. We're going to try to do one of these videos maybe every day. I don't know. Maybe I'll do a couple more tomorrow, and then I'll eventually post the whole series. So part one. So stay tuned for part 2. All right. Bye, everybody.